Hi, everybody.
Heyo.
Heyo. So I have all the videos for chapter 21 ready. Something is going on with my internet. I'll upload them as soon as I can. They'll be up tonight at some point. But we got to start going through chapter 21 now, which is fine. So you'll get 2 treatments of it, one in the videos, and then we'll do a little bit more practice with some of the parts of 21 together.
Are we not going in line of like going 19, 20, 21?
If you look at the order of chapters in the suggested schedule, 21 is the next chapter that we're covering.
Well, shit, I'm printing the wrong one.
There's a reason for that also. It's because in terms of us learning about biomolecules, looking at proteins first right after we finish talking about amines makes the most sense. So we've just learned about formation of amide bonds between amines and carboxylic acids. Guess what the most important part of the chemistry of proteins is? The formation of amide bonds. So that's why we're doing this. Now, the other thing is we're probably going to do more with proteins than any of the other kinds of biomolecules. The reason why is intentionally I'm making the class light on whoever has background noise happening. Can you mute yourself? Ariana, can you mute yourself? I don't know if it's quiet where you are. I'm still getting background. Somebody else. All right, it went away. That's awesome. That doesn't mean that you can't unmute yourself if you want to ask a question. Please do. All right, I love being interrupted, just not with background noise. Because pretty much everybody that's in this class is going to take Bio 160 also. You're not going to, Orlina. That's why I said pretty much. There's always an exception someplace.
This is my last class for my thing, which I got accepted to the hygiene program that I was going for. So
did you ever
excited.
take Bio 160?
I don't know, maybe.
I
yeah,
bet
biology.
You did.
Yeah, biology 160. Yeah, I took that.
General biology?
Yeah. I took that a while
so
ago.
Either you have taken general biology at some point or you will take it at some point. And in that class, you learn a lot about nucleic acids. So you learn about the genetic code, you learn about the DNA double helix, you learn a lot about translation and transcription. So we don't need to cover all that for this class. We're focused on the chemistry of different biological molecules. That's why the biochemistry part of this class. So if it was a majors level biochemistry class, we would learn quite a bit about the chemistry of nucleic acids. But honestly, it's kind of complex and it's very relatable to the chemistry that we're going to learn when we cover proteins in depth and also do a lot with carbohydrates and lipids. All right, so we'll go light on nucleic acids, but that's by design because we just don't have time to cover everything. Also check the tentative schedule because I have shifted due dates just a little bit. Chapter 21 was supposed to be due in a couple of days. We're just starting covering it right now, so it's going to be due a week from today. All right, that should give you guys plenty of time to get that homework done. Yeah, I try and keep due dates for things reasonable. I think that 2 days after beginning a new chapter is not a reasonable due date for the homework on said chapter, so we adjust, and that's totally fine. If you notice the last week of the schedule, we have time where it just says like exam 3 review. And honestly, based on the way our class is scheduled, we could have meetings on both Monday and Wednesday of that week before we have our exam. So there's nothing that says we can't do our final exam on Thursday of that week. And that would be perfectly fine. So we could have 2 meetings that week and would have plenty of time. Orlina, question.
So after we're done with our exam, about when would our grades be posted? I have to get my actual
They have to be in the following Monday, I think.
Okay, so our last day is
I can give you the date. Hold on just a second.
like the 19th, I believe.
Hold on just a second. I think the grades will certainly be in by the 22nd. That's the date that the college requires them to be in.
Perfect, because I have to have mine in by the 26th, so.
Okay, sweet.
Perfect.
So anyway, that's what I got for you. I'm not saying that they won't be in sooner than that, but if we're taking our exam Thursday of exam week, which is great because it gives us the most possible time to study before the 3rd exam, then you got to give me a couple of days to get those grades turned around. The other cool thing is for the 3rd exam, you will earn your curve on the exam for free. You don't have to do corrections because like I don't want to get more assignments from you right up until the grades have to be in at the college. So for exam 3, you're gonna get your curve for free. For exam 2 that's coming up on Friday, we'll do exam corrections for that one the same way we did for the 1st exam. So I'm going to try and go through the biochemistry chapters using a mix of stuff that I will write on the overhead and the PowerPoints for the chapter. And the reason why is because the biochemistry chapters are chock full of drawings and I can't like redraw everything on the overhead. It would just take us like 6 years to cover those chapters. So we'll highlight and look at parts of the PowerPoints together. It'll basically be like an expanded version of the topical videos. Oops. And I will try and highlight in the PowerPoints slides that I don't think have much use at all. All right. There's always some extra fluff in the chapters of an introductory textbook because we want to connect everything to something that you understand, which is good, but also like it's hard to always realize where I'm conveying important information and where like this is stuff that you really don't need to remember to do well in the course. So I'll try and be as clear as I can about what parts of the PowerPoint are important and which ones aren't. So what is a protein? Proteins are what we get when we join individual amino acids together. So an amino acid is named amino acid because it has an amine functional group and a carboxylic acid functional group together on the same molecule. We get proteins by putting lots of amino acids together with amide bonds. So amide bonds in the context of forming proteins are also called peptide bonds. So those bonds shown in red are what we get when we put the amine of one amino acid together with the carboxylic acid of another carboxylic acid and do a dehydration to attach the carbonyl carbon to the amine nitrogen and make an amide bond. This second slide, like, who cares? All right. Dietary protein, you can certainly get plenty of protein without eating meat and milk. So this slide was from an author who had a very specific, not vegetarian friendly view of how proteins are consumed. We now know that you can get plenty of protein without ever touching animal proteins at all. All right. So if you choose to do that, then that's awesome. We don't store proteins, generally speaking, and proteins don't last that long. So depending on what type of protein, it probably has some kind of a lifespan in the region of minutes to hours, which means that if we have a protein that has an important job, we have to keep making more of that protein all the time, which means that we have to consume the parts of the protein, the amino acids. We have to be consuming or synthesizing those all the time to make more copies of those proteins because they don't last that long. All right. So this is a pretty important slide, not because I want you to memorize all of these different specific proteins, but because it's important for us to realize that proteins have some important jobs, all right? So what jobs, if I was to distill what I meant about important jobs of proteins, proteins can be transport proteins, so they carry something someplace, all right? Hemoglobin is an excellent example of that. There are also lots of other important transfer proteins that transport other molecules. So hemoglobin transports oxygen. You've probably heard of that one before. So transport is an important function of proteins. Structure is another really important job that proteins have. So like giving, you know, a three-dimensional rigidity to parts of the cell or to parts of organs and tissues. So collagen and keratin are both important structural proteins. Collagen lending itself to the structure of skin, keratin to the structure of hair, all right? Ferritin is another storage kind of protein. So besides transport, you can also be important for storing some kind of molecule. So ferritin stores excess iron. Myoglobin is very similar to hemoglobin, but it doesn't transport oxygen. It just tucks extra oxygen away for when we need it. So myoglobin is the storage version of hemoglobin. It stores oxygen instead of transporting it. And then actin and myosin actually sort of have both structural and like signaling kinds of roles. Insulin also a signal. So signal meaning like the body needs to tell some other part of the body to do something, and proteins are involved in that signaling very often. And then finally, probably the most important of the jobs isn't directly anywhere on this page, but catalysis. So enzymes, most enzymes that are known are proteins, and enzymes work to catalyze chemical reactions. So if I had to make a short list of important jobs of proteins, my list would probably be something like those 5 things: structure, storage, transport, signaling, and catalysis. That I think covers like a vast majority of the jobs that proteins have. That's reductive because that's pretty much everything that's important to keeping living things living. All right. So we have to have like structure to be able to set up space in which to keep our cell stable, right? We have to be able to store molecules for use later. We have to be able to move molecules around to the places where they're needed. We have to be able to signal biochemically for when a change in the way the organism is functioning is happening. And we have to be able to make chemical reactions happen really fast. So all those are essential jobs of living things. Proteins do all of them. All right, so proteins are super important. So proteins are made up of amino acids. So we're going to start in our chapter 21 with the structure of amino acids. And there are 20 different amino acids that are commonly found in human proteins. In reality, it's more like 21 or 22, but the 20 are really common. And then there are a couple of like guest stars that show up once in a while. The reason why the guest stars only show up once in a while is because they don't have specific codons that code for them in translation. So we're only going to worry about 20 amino acids because those are the ones that are coded for in our genetic code. When we take an mRNA and turn it into a protein through the process of translation, that's not like a chemical synthesis. That's we use the mRNA to assemble a protein. And we're not going to talk too much about that because that's Bio 160 stuff. I just sidetracked myself. Those messenger RNAs only can code for 20 different amino acids. So those are the 20 that we're going to focus on. All right, so an amino acid is made up of an amine group, a carboxylic acid group. Now, the carbon in between those two, we refer to that as an alpha carbon. All right, it's just a nomenclature thing so we can keep straight what carbon within an amino acid we're talking about. And then attached to that alpha carbon normally is a hydrogen and then some other group. So this R group is what gives the amino acid its identity among those 20 amino acids. They're all identical except for differences in that R group or side chain. So the simplest amino acid is glycine, where its R group, its side chain is just a hydrogen. All right, that's kind of anticlimactic. And sometimes the R group contains a basic nitrogen atom, and we call those basic amino acids. Sometimes that R group contains an extra carboxylic acid functional group in it. We call those 2 amino acids acidic amino acids. So I'll show you structures of those. You do not have to memorize the structures of all 20 amino acids. This is not a senior level biochemistry class. If it was a senior level biochemistry class, you would have to memorize the structure of all 20. All right, it's really not that hard. You just make note cards. So another important thing for us to realize about the structure of amino acids is that when amino acids are by themselves, when they're not put together to form peptides, the amine and the carboxylic acid look a way that we have seen before in earlier chapters. Remember that amines are basic functional groups, which means that they can pick up an extra proton and have a +1 charge. Remember that carboxylic acids are acidic functional groups, which means that they can give away an H+ and end up with a negative charge. It turns out that most of the time, when an amino acid is in neutral solution, both of those things happen to it. So amines become NH3+, carboxylic acids become carboxylates, COO-, and this is what an amino acid looks like in aqueous solution. So they're neutrally charged overall, generally speaking, but they have 2 different charged atoms on them, the nitrogen and the oxygen of the amine and of the carboxylate. So we call this form of the amino acid, where the nitrogen has a positive charge and the carboxylate a negative charge, we call it the Zwitter ion form. I think that's like one of the coolest words ever in the history of the world.
Where did that word come from?
I don't know. Yeah, I've never looked at the etymology of it, and nobody's ever asked about it before. I can do some research and figure it out if you're interested. So
It's
I
probably
normally
somebody's last name.
So based on the ZW, it's almost certainly from a German root.
It's all the crazy overseas scientists that come up with the cool things. They're like, this is going to be considered the Zwitter ion.
Germany has like a murderer's row of important historical chemists. Yeah, Germany is like mad props to the Germans for bringing out chemical theory, right? So you can even like hear, you know, with the German accent, like the Zwitter ion, right? So you can that's definitely from a German root, but I'll figure out where that word comes from, and we can talk more about that later. Nobody's ever asked. I don't know. So if you were interested in the structures of the different amino acids, here are most of them, all right? So these amino acids are the amino acids that are neutral in aqueous solution. So what that means is their side chains don't carry a negative or a positive charge in normal aqueous solution.
Does the star indicate it's an essential?
Yep, you can even see that at the bottom there. We're just going to talk about that in a second. So the amino acids that are starred are essential amino acids. All that means is that our bodies cannot make those amino acids. We have to consume them in our diets to get them. And I'm not a biologist as much as I am a chemist. Off the top of my head, I never remember which of the amino acids are essential and which ones aren't, because like that makes no difference to their biochemistry to me at all. It's just a dietary thing, right? Some of them I have to eat and some of them my body can make if I don't have enough. So there is one interesting one in here. So phenylalanine. If you guys have ever heard of phenylketonuria, have you ever heard of that? It's a disease. any excess phenylalanine that somebody with that disease eats is incredibly toxic because they don't have the enzymatic machinery to get rid of excess phenylalanine. So their phenylalanine in their diets has to be like incredibly carefully monitored because phenylalanine is an essential amino acid. You need some of it, right, to function. But if they have like a little bit too much, it's toxic. So that's not a disease that you want at all. Notice that the amino acids have a number of different functional groups in them. So asparagine, for example, its side chain contains an amide functional group. Cysteine, a really important side chain, contains that SH group, a thiol functional group. So cysteine is going to be really important to us when we start to talk about how proteins fold up. We're going to talk about something called disulfide bonds that hold different distant parts of proteins together. And that happens because two cysteines at different places on a peptide can form a covalent bond using their sulfurs. So you do not need to memorize all of these, all right? You will open book, open note for the third exam. So you'll have, I would highly suggest that you print out a page that has all 20 of the amino acids on it so you can see those structures as you're taking the test. So the last of the amino acids are the ones with charged side chains, the ones that are acidic or basic amino acids. We probably should do just a little bit more than just neutral and charged. So I'm going to really quick draw the 20 amino acids over again. All right, but I'll draw them in bond line drawing form because that for some of you will be easier to interpret. So the important thing for us to look at in the acidic amino acids is that those two side chains, the side chain of aspartic acid contains a COO minus, a carboxylate. So that's a carboxylic acid functional group. It just has lost its proton, just like the one that is part of every amino acid. And glutamic acid, also a carboxylic acid functional group. So the key for all three of the basic amino acids is they have a nitrogen in them that can pick up an extra H+. So for arginine and lysine, those nitrogens are shown that have already picked up the extra H+. For histidine, that nitrogen right there can pick up an extra H+ if the pH is low enough. So all three of them are basic amino acids because their side chains contain a nitrogen that can pick up an extra proton. That's the important thing to remember. Give me just a second, y'all.
For everything except for, I don't have them memorized yet. Everything except for glycine and the one with the other carboxylic. Never mind. Everything except for glycine that has the chirality center.
Everything except for glycine, what now?
Everything except glycine would have a chirality center, right?
Chirality center, yes, it would. You don't have to worry about the chiral center in it too much. We're going to talk just a little bit about that, all right? Just a little bit about that.
Okay.
So, okay. So what I'm going to do is I'm going to take these amino acids and I'm going to redraw them as bond line drawings, and we're going to try and sort of organize them in a little bit more intelligent a way than this slide organizes them, because there's something important about their structures. Some of them have polar side chains and some of them have non-polar side chains. Now, that's really important for us to remember, because when we talk about how proteins fold, whether the side chain is polar or non-polar makes a really big difference. All right, so let's try and draw our amino acids with non-polar side chains first. And I'm going to draw them in bond line drawing format. So glycine doesn't have a side chain, or rather its side chain is just a hydrogen. So that carbon in the middle right there is our alpha carbon, the one that exists in between the amine nitrogen and the carboxyl carbon. All right, that carbon in the middle is our alpha carbon. So glycine is nonpolar because hydrogen bound to carbon is a nonpolar bond. And then alanine is a little bit bigger than that. So what I'm going to show you guys is that the side chain, the R group that is coming out from that alpha carbon, because of the stereochemistry at that carbon right there, that R group always points toward us. So I'm showing it with a wedged bond to that CH3 right there. All right, so that's alanine. Now I could show the two hydrogens that are bound to that glycine like this and this, and I could show that that hydrogen is still back there, that fourth bond to that carbon on alanine, but we don't have to show our hydrogens in a bond line drawing. So I'm leaving those out because it makes the drawings a little bit cleaner. All right, so can you guys look through your list of the amino acids and think about which other side chains would likely be non-polar?
Isoleucine.
Isoleucine is a little bit bigger one. All right.
Leucine both then.
Yeah, they're both. They're both. All right. So leucine has kind of like a V at the end, and isoleucine splits a little bit earlier. So isoleucine looks like this. And leucine like this. So as I'm drawing, compare what I've drawn in my bond line drawing to what you see over here, and try and make sense of how that condensed structure looks when it's translated into bond line drawing. To me, the bond line drawings are a lot easier to interpret in terms of seeing that things are polar or non-polar. All right. Now, proline is another example of a non-polar amino acid, but it's a weirdo. So it's weird because the nitrogen part of proline is actually attached to the alpha carbon through a chain. So I'll show what that looks like. So if you want to, you could also draw out these COO minuses like this. All right, so there's your carboxylate group. Now the proline has a 1, 2, 3 carbon bridge that actually forms a covalent bond back to that nitrogen. So that does something really important to the protein chain when proline gets incorporated. So we'll talk about that because proline is a really important structural component of proteins. Can you see any other non-polar side chains? I see at least 2 more. Phenylalanine looks a lot like alanine, but with an extra phenyl group or benzene ring attached to it. So all of that side chain is just CH bonds. So of course, that's going to be a nonpolar side chain. I see one more. Valine, which resembles leucine, but its side chain is shorter. So we get a branch that comes out right from the alpha carbon. So those are our nonpolar side chains. All right. We might consider tryptophan also to be a nonpolar side chain, just because even though it does have that NH group on it, the bulk of it is nonpolar. All right, those two rings are almost all carbon-hydrogen bonds. So tryptophan is mostly nonpolar, so we could kind of put it anywhere. All right, not that important one way or the other. So tryptophan, I would consider that mostly nonpolar. Remember, polarity is not always a yes or no, but like a sliding scale. All right. So let's move on and now talk about some polar side chains. All right.
Can you move that back for just a second?
I can.
Okay, thank you.
You're welcome. So one thing that we'll get to in the slides, but I want you to realize right now is that for all the amino acids, there's something special about the way that their atoms are organized in space, where the hydrogen that's attached to that alpha carbon is always pointing backward, and the R group is always pointed forward when our amine group is on the left and when our carboxylate is on the right. So when Ariana was talking about the chiral center or the chirality of the amino acid, they're talking about how the four groups are all attached to that alpha carbon right there. And it turns out that for amino acids that are used in living creatures on Earth, almost always, whenever we have the ammonium group on the left and the carboxylate on the right, the R group is going to be pointed out toward us. So that's called an L amino acid. That's just the organization of those four groups, the way that they're attached to that carbon. Orlina, did you get what you need out of this list now?
Yeah, pretty much.
Okay.
If anything, I'll go back and just look at it later.
You sure can, or you could do it yourself. You could take these and turn them into bond line drawings, and it's good practice to do that, all right? So for the polar side chains, let's look at our list. As we go down, asparagine definitely is polar. So note that asparagine actually looks a lot like one of the charged amino acids, aspartic acid. So asparagine has a primary amide functional group on its side chain. And glutamine, I'm going to do next because it also has a primary amide functional group attached to its side chain. The only difference is the side chain is one carbon longer. We already talked about cysteine being one of the polar side chains. that has a thiol functional group on it. Serine looks almost identical to cysteine, except instead of an SH, it has an OH, an alcohol for its side chain. Threonine looks similar to serine, but it contains an extra methyl group. So we have done lots and lots of talking about different functional groups throughout the organic part of the course. Now that we're into the biochemistry part of the course, you can see lots of those functional groups show up, right? The amide functional group shows up in this side chain in this one, the thiol functional group, alcohol functional groups in serine and threonine, and even like a phenol kind of functional group shows up in tyrosine. So tyrosine looks a lot like phenylalanine, but with an extra OH group attached. Just making sure I haven't left anything out. Oh, you know what, Orlina, you probably were wanting to go back to this page to put one more nonpolar amino acid in. I think methionine qualifies best as nonpolar. So the functional group in there isn't one that we've really looked at very much. I want to think for just a second about how we might name that functional group, because I bet you guys can come up with a good name for it. So you can see that we have two CH2s and then a sulfur and then another CH3 here. That is what methionine looks like. If that sulfur were an oxygen instead, what would we call that functional group? An oxygen with two carbon-based groups attached to it.
Ether.
Yep, an ether functional group. So with a sulfur instead of an oxygen, we call that functional group in methionine a thioether. So remember that ethers make a molecule polar because the 109.5 degree bond angle between our two carbons here and the bent shape around oxygen would mean that we'd have a delta negative on oxygen and a delta positive on the carbon side. The same thing is true with sulfur, but sulfur and carbon are a lot closer in electronegativity. So a thioether doesn't really make the functional group that polar. So the side chain here is just not polar enough for us to really consider it to be polar compared to the others. Remember, it's not a yes or no, it's kind of a sliding scale. So methionine and tryptophan obviously are more polar than something like phenylalanine is, that doesn't have any polar bonds in the side chain at all. All right.
Is it a thioether or is it called a thioether?
It's a thioether. In other words, methionine is an example of the functional group thioether.
Okay.
Does that make sense?
Yeah, just double checking.
Yeah. I just didn't want to write an example of the functional group thioether because that's too many words to write down. Okay, so we've gotten to nonpolar and polar side chains. From there, let's move ahead on our slides and look at the charged side chains. So we'll do the bond line drawing versions of those. So aspartic acid or aspartate, depending on whether that thing has its proton or not. It's called an acidic side chain because it has a carboxylic acid functional group on its side chain. Notice that that's also a carboxylic acid functional group. I've just shorthanded it for time's sake. All right. So aspartic acid, when it's by itself, has 2 COO minus groups on it, which means that the acidic functional groups lose protons and tend to be negatively charged functional groups in aqueous solution. So just like aspartic acid, glutamic acid also contains a carboxylic acid functional group in its side chain, and we can call that glutamate if we're talking about it after it's lost its proton like it normally exists in solution. So the only difference between aspartate and glutamate is that glutamate has 1 extra carbon in its side chain. So if you look at glutamate and glutamine, the only difference is carboxylic acid on the side chain versus amide on the side chain. They're very, very similar in structure. And then we've only got a couple more left to go, actually 3 more left to go, the basic amino acids. So lysine is a fun one to draw because it's got a really long side chain. If we count up all of those CH2s in the side chain, there are 4 of them. So there's 1, 2, 3, 4 carbons, and then after that, a primary amine, which in aqueous solution tends to have an extra hydrogen and be positively charged. Now, arginine looks similar. We're not even going to talk about what that functional group actually is called for arginine side chain, but it contains 3 different nitrogens, one of which can have an extra proton attached to it. So that nitrogen right there is the one that we're talking about that can pick up an extra proton and be positively charged. And then finally, histidine. So I'm going to show histidine after it's picked up an extra proton and is plus charged. So in other words, when this nitrogen has 1 more proton attached to it, just so you see it both ways here. One, two, three. Four, 5. So something like that, if that nitrogen had an extra proton attached to it, it would have a +1 charge. So now you have both versions, the bond line drawing version and the condensed structure version of each one of the 20 amino acids. It's important for us to organize them into polar and nonpolar, and charged are like the ultimate in polar, right? They carry a whole charge on the side chain. The reason why has to do with how proteins fold, and we'll get back to that later on. But remember that we took time to organize them into polar and nonpolar because it's going to be important for us to talk about a little bit later. So you don't have to know everything about the stereochemistry of amino acids, but remember the way that I've drawn them. If our NH3 plus is on the left and our carboxylate is on the right, then our side chain, our R group, is always pointed out toward us. All right, that's the way that all natural amino acids look. So those are the L isomer of the amino acid. Remember also that amino acids are neutral at pHs, well, they tend to be neutral at pHs close to physiological pH, so somewhere close to 7. That's not true for the acidic or the basic amino acids, all right? And it's not always true for the amino acids anyway. So amino acids change their structure depending on the pH. So let's take a look at that for just a second, all right? And we'll use alanine as an example of this. So I'm going to do this drawing instead of on the PowerPoint because it's more fun that way. Holy crap, we just toasted an entire notepad. I had more of those someplace.
Jess, do you have all of these memorized?
Me?
Yeah.
Oh, of course.
Okay.
But only because it's important for us to be able to talk about them individually, because as we get further into biochemistry, there are more and more things that we have to know about the chemistry of specific side chains. Like they have certain jobs that are important for them. For example, you wouldn't think that glycine would be that important because it just has a hydrogen as a side chain, but the lack of a big bulky group there is actually really important to the structure of lots of proteins. So whenever we incorporate glycine into a peptide chain, that allows for the peptide to have a little bit more freedom to wiggle in the chain at that spot because there's nothing sort of preventing too much movement. We'll talk about more examples like that later on. But all 20 of the amino acids have like little quirks about where they like to show up in proteins that are worth knowing about if you are an enzyme biochemist, for example. All right. So learning those 20 amino acids is like a first step, but the more important thing is learning the quirks about why they do what they do when they're incorporated into proteins. We don't have to know all that stuff for this class, that's for sure. So let's, instead of doing alanine like they do in the PowerPoint slides, let's look at valine instead. So the important thing here is I want to pick a side chain that is nonpolar. So we're not talking about a side chain that can gain or lose any protons here, certainly. All right. So this is what, and I'll even draw out the carboxylic acid from here on out. But this is the zwitterion form of valine. So at pH is close to 7, all right? So around 7-ish is a physiological kind of pH, all right? pH of blood or something like that. So within 1 unit, certainly, that would be the pH of blood. If we're more than a unit off from that, then we'd be in bad shape. Let's take a look at the difference when we take this valine and put it in a different environment. So what if our valine was present in a solution that had a pH of 2 instead? So from the end of Chem 121, the pH scale, we know that the lower the pH scale is, the higher our concentration of H+. So at a very low pH, we have lots and lots and lots of H+. So in an environment where there are lots and lots and lots of H+, there will be no reason to not have the H+ attached to that nitrogen, but the carboxylic acid functional group, if there's lots and lots and lots of H pluses around, that carboxylic acid is also going to have its proton attached. So at a low pH, a pH of 2, a neutral amino acid like valine, or a valine which will be neutral at a pH of 7, ends up with a net positive charge because our carboxylic acid functional group now has a charge of 0 and our ammonium a charge of +1. So valine becomes positively charged at low pHs. Well, what would happen to valine at a high pH, like a pH of above 12? So at a pH of 12 or 13, our concentration of H plus is very, very low. There's almost no H pluses floating around in solution. So what do we think our two functional groups are going to look like when there's very little H plus around? Instead of our nitrogen having an extra proton and a plus charge, when there's not very much H plus around, our amine is going to be uncharged. It's going to have just its 2 hydrogens, but not have picked up an extra H plus. At a pH of 13, our carboxylic acid functional group will also have lost its proton and have a -1 charge. So look at the net charge of valine at a high pH. At a high pH, that functional group has a charge of 0 and the carboxylate a charge of -1. So we have a net negative charge if that amino acid is present in a solution with a very high pH. So that doesn't mean that this is valine and these two aren't valine. All 3 of these are valine. The important thing is that valine can change what it looks like based on what pH the solution is that it's in. All right? We know that because we know that amines are basic functional groups. They can pick up extra H pluses when there's enough H plus around. All right. We also know that if there aren't enough H pluses around, carboxylic acids can give H pluses away. So when does a carboxylic acid have its proton? When there's lots of H plus around, when pH is low. When does an amine pick up an extra proton? Well, anytime that the solution is neutral or acidic. When do amines not pick up a proton? Well, when there's not very much H plus around, when there aren't very many protons. And of course, if there aren't many H pluses around, the carboxylic acid will have lost its proton when there aren't very many around. So that's how we can tell what those functional groups are going to look like at different pHs. Does that make sense to everybody? We can get even a little more complex than that. So for example, what about lysine? What do you think lysine would look like at pH 2? What about at pH 13?
So I understand all 3 of those are still valine. Does it ever get some nomenclature added to it to distinguish when it has?
Nope. And the reason why is because when we're talking about the amino acids in proteins, they're not free amines and free carboxylic acids anymore. This would be part of an amide functional group, and this would be part of another amide functional group. So this behavior only happens to amino acids when they're floating all by themselves. All right. And most of the time when they're floating all by themselves, they're in some sort of a solution that's pretty close to physiological pH, like pH 7. Could you think of a place where you might actually find an amino acid at a low pH? There's one that I can think of pretty easily, like a physical location. Go ahead.
The stomach is a pH of, in humans, is like 1 to 3, isn't it?
Yep. So stomach acid is very acidic solution. So if we ended up with individual amino acids in the stomach, then they might end up with net positive charges like this because of the pH. All right. So this is just something for us to remember that's connected to chemistry that we already know. We already know that amines are basic functional groups. They can pick up an extra proton. We already know that carboxylic acids are acidic functional groups. They can give a proton away in places where there aren't many protons around.
So quick question on that. So if you find this in the stomach, obviously where Ariana said that it's a 1 to 3, would that be the portion of why it starts to break down there, is because it has the hydrogens then added onto it, which then as it gets digested, it gets like broken down?
So normally the amino acids themselves do not get broken apart in the stomach. The peptides, proteins get broken, their amide bonds get broken, and that's partially due to the acid in the stomach. Remember that we talked about in chapter 17, amide hydrolysis can happen in aqueous acid, right? So aqueous acid can cause breaking of amide bonds. The other thing that really helps a lot is the presence of enzymes that catalyze that reaction and make it happen faster. So that's the biggest part of why proteins break apart into amino acids so fast in the stomach, is because of these enzymes called peptidases that catalyze amide bond cleavage.
That's pepsinogen that gets changed into the enzyme in the presence of the acid, right?
Yes, that's right. Another thing that we don't have to know for this class, but that is an example of something that we call a proenzyme, an enzyme that needs to be activated through cleavage in order to work. So we talk about those just a little bit at the end of this chapter, but I didn't even give an example. So you just gave one for us. Pepsin's a good example. So lysine. At a pH of 2, the amine functional group will have picked up an extra proton and have a +1 charge. The carboxylic acid functional group will have its proton and be neutral. And what about the side chain? Remember, the side chain on lysine is another amine functional group. Well, what do amines do? In acidic solution, they pick up an extra proton and have a +1 charge. So at a low pH, a pH of 2, lysine would have a net +2 charge. At a pH of 13, lysine would end up with a net -1 charge. Now at a pH of 7, something close to physiological pH, lysine would end up with a net +1. And the reason why is because the Zwitterion form of lysine has a plus charge on the ammonium, a negative charge on the carboxylate, but our side chain breaks the tie here. The amine carrying an extra proton gives lysine a net +1 charge at physiological pHs. So that's why lysine is important as one of the charged side chain amino acids. It carries a plus charge as a side chain at physiological pH. So that is kind of what this section of the chapter is talking about when it talks about how amino acids look different at different pHs. All right.
So when they form their peptide bonds,
The only
that
charges that are left are the charges in the side chains, if there are any. Yep.
are the charges affected by pH when they have peptide bonds attached to the individual amino acids? Like when you have before it's a protein, when it's just a polypeptide, a series of amino acids, are those side chains of each individual amino acid, are they affected by pH the same way when they're
Yes.
a polypeptide?
The side chains are affected by pH the same way that the
Amino acids?
individual amino acids would be.
Okay.
It's just
So
that the ammonium and carboxylates of the amino acids, the functional groups that aren't part of the side chain, they're not there anymore. They're all part of amides, so they're not charged any longer. So we'll get to that.
Lysine, which has a side chain with a nitrogen in it, if the protein was unfolded in an acidic environment, that's when it would be the same.
I'm not sure what you're asking, but I guarantee you that your question will be answered as we traverse through the chapter.
Okay.
Okay. And if it's not, we can revisit it a little bit later. So we haven't even talked about formation of peptides yet, but these ammonium groups and carboxylate groups that aren't part of the side chain, but part of just individual amino acids, those get converted into amide functional groups when peptides form anyway. So let's start from this slide. Okay, so we've already talked about this. Let's move on to what happens when those individual amino acids get together to make peptides. All right, so individual amino acids use the ammonium group of one amino acid and the carboxylate of another and form an amide bond, just like we learned about in chapter 17. All right, we just happen to call these peptide bonds when they're part of a protein. All right. So notice that the carboxylate here is not a carboxylate anymore. It doesn't have an O- attached to it. So we can't carry a charge here anymore after that peptide bond forms. That nitrogen is just an NH that's part of an amide now. It cannot carry an extra plus charge anymore as part of that amide functional group. So the only things left in the peptide that can carry charges are the very first amino acids, nitrogen, and the very last amino acids, carboxylate, and then if we have any side chains that have charges. Those are the only charges left over once we form all of the peptide bonds in a peptide. So here is a tripeptide, in a dipeptide meaning 2 amino acids joined together, a tripeptide meaning 3 amino acids joined together, and then sometimes small peptides can be used for signaling purposes that are only 6, 8, 9 amino acids long. Sometimes very small proteins can be as small as only 40 amino acids big, and proteins can be up to like 1,000 amino acids big at the biggest. All right, maybe even larger. I'm not. How many amino acids are in the ribosome? Proteins can be really big. All right. I don't know the upper end of the number of amino acids that can exist in a protein, but I can definitely look that up. It's just not popping into my head right off the bat. So I want you guys to be able to draw what a peptide looks like when we join individual amino acids together. So we do that through a reaction that we're familiar with called dehydration, where we take an oxygen off of the carboxyl group and 2 hydrogens off of the ammonium group and end up attaching the ammonium nitrogen to the carbonyl carbon and creating a new amide bond. All right, so let's skip ahead a little bit and try and draw a tripeptide. So I'm going to have you guys look back in your notes. Let's do not alanine or serine. So let's do methionine. Glutamic acid, and then lysine. So try and draw me a tripeptide with that sequence. Methionine bound to glutamic acid, bound to lysine. So always the order that we draw our peptide sequence in starts with the NH3 plus side and ends up with the carboxylate side. So we often refer to this as the N-terminal amino acid and this as the C-terminal amino acid. So we start with the ammonium group of methionine and then move through the sequence that way. Let's take a second, see if you can figure it out. So if you're having issues, try and draw all three of them individually first. And we're going to assume that this tripeptide is existing at a pH of 7, all right? Very close to physiological pH. So our carboxylate should be deprotonated and our ammonium should be protonated for those two side chains. Okay, so the atoms that we're going to be linking together are that carbon to that nitrogen and that carbon to that nitrogen. There's our carbonyl of that carboxylate. We've taken the oxygen off. We're taking 2 hydrogens off of that nitrogen, and we're left with an NH that belongs to our glutamate. So the side chain of our glutamate is now going to be pointing downward. So since our side chain is pointing downward instead of coming out at us, it's going to be going backward instead. So think about this second amino acid as being sort of flipped over 180 degrees like a pancake from where this first one is. And then we have our carbonyl group of our second carboxyl here. And we're going to attach that to the nitrogen of our lysine. And that finishes off our tripeptide. Now, if you want to draw them the way the PowerPoint does with more of a condensed structure vibe to them, that's fine. I really like this way because it takes up less space. It takes like 1/10 the time to draw the bond line drawings as it does to draw condensed structures. So we just have to remember that any amino acids that have the alpha carbon pointing downward, those R groups are going to be pointed away from us instead of being pointed out toward us. All right. And if you want to just flatten everything out and just make that a regular bond line and that a regular bond line and that a regular bond line, that's fine, I suppose. I'm just trying to like leave some element of stereochemistry left in the class, not completely scrub it out.
Do you have to put that second one down, or can you have them all facing up?
You can't have it all be pointing up because that bond line doesn't make any sense if we draw like remember our bond angles around here. Well, we don't have to talk everything about that, but the bond angles around that bond are something actually close to 120, which is a little bit unexpected. It has something to do with resonance. The bond angle here is 109.5, 120, 120, 109.5. So really the best way for us to draw this is like a zigzag. And in the zigzag, after this nitrogen, the next bond has to point down, which is why we want to have this R group coming down instead of going up, which doesn't really make very much sense in the drawing. If the zigzag points down, then whatever's coming out of here should be pointed down, not up. Does that make sense?
Yes, it does. It is important. Okay.
If
Okay.
you accidentally drew the glutamate, the middle one,
Yep.
if you accidentally drew that with a solid line as opposed to going away but coming at us, that would be changing it from an L
to a D amino acid. Yes.
Okay.
Yep. Which would make it unnatural. All right. So no organism would have a glutamate with a side chain that pointed out toward us in this orientation. That would be an unnatural amino acid. As a matter of fact, just as a joke, for a while, well, it wasn't really a joke. It was a serious idea, but it was one that turned out to be too difficult to actually do. I'm going to stop screen sharing just so I can see you guys for a second. So when I was in grad school, my advisor and I came up with this crazy idea that we wanted to try and create an organism that functioned as a mirror image of like other organisms. So we wanted to make a strain of E. coli that functioned entirely with D amino acids instead of L amino acids. And instead of having regular DNA that was a right-handed helix, we would try and have artificial DNA that was used for its genetic code that was a left-handed helix instead. So we created this idea of basically a mirror image of the normal biochemical process in this small organism. Now, it turns out that that's like way too hard to actually implement, but the nickname that we gave to the E. coli were D. coli because they use D amino acids instead of L amino acids. So anyway, basically every natural organism that we know about uses L amino acids for almost everything other than for some very specific jobs that aren't part of their bulk proteins. So L amino acids are where it's at in terms of biochemistry.
So from your picture, I'm getting a little confused. How did you get that double bond to oxygen right there?
What double bond to oxygen? Are you talking about? Oh, sorry. My screen's not shared anymore. Give me a second.
You're muted, Orleana.
I was trying to ask my question before everything went away, but then sidetrack tangent happened.
So where are you looking?
So like right after the first MET one, it comes down and it's into a double bond into oxygen. But if I'm looking at this, it right there to me
This
is where
here is this carbon. So that COO minus, if I draw methionine out, the first amino acid looks like this when it's by itself.
okay. Yeah, now I get it.
So COO minus is the abbreviation for a carboxylate. So the carbonyl group bound to an O minus.
I get it now.
It's a good question. It's an important question.
Well, as I was drawing it, I was like, wait a minute. I'm so confused all of a sudden, but now understanding that okay, perfect.
So we can also draw the structure without all of its three-dimensional glory and beauty just by doing sort of a modified condensed format where we have our NH3 group and then our alpha carbon with a hydrogen and the R group. So in this way of drawing it, all of the R groups would point right down from the chain, but you don't see the three-dimensional structure at all that you see with our bond line drawing form. So I think the bond line is a lot prettier and more fun to draw.
And
That's
as you
just-
said with that, we don't have to, like when we're doing it, we don't have to actually put like that extra little like black line in there to show that it's facing.
You don't have to
off
show
of the-
wedges and dashes if you don't want to.
Okay, because I think for me, that would probably be a little bit more confusing thinking that there might be like an extra carbon in there.
But all that wedge shows is the direction that that bond is traveling. So that just means that this bond is coming out at us a little bit. I'm just trying to help. I'm not trying to convince you to not flatten it out. So the important thing that we sort of talked about but didn't completely talk about is that if we combine two amino acids, we could combine them in two different orders. So serine first and then alanine. So remember, we talk about in terms of the sequence of amino acids in a peptide, we talk about them in order where we start with the one that has its amine sticking out. The N-terminal amino acid is first, and the C-terminal amino acid, the one with the free carboxylate, is the last one in the sequence. It's really important if we're shorthanding a sequence out. So met, glue, lyse are 3 amino acid peptide. The methionine was the one that was the N-terminal amino acid, and that's like always written in that order. So if we're writing a peptide sequence, the N-terminal amino acid is the one we write first. And that's what this slide says, just like I said before I got to it. So we've already covered how to draw a tripeptide, which is even bigger than a dipeptide. I would expect you to be able to draw a tripeptide of any 3 of the 20 amino acids. You don't have to memorize all 20 structures, but you do need to be able to use them to draw a tripeptide. So here's one of valine and glycine. Remember, we're doing a dehydration, so the carboxylate oxygen of valine is going to go away. 2 of those hydrogens on glycine are going to go away, and we end up with this nitrogen attached to that carbonyl group to make our new amide bond. If you want to draw them like this in a straight line, I suppose that's okay. It's really boring. I think you guys are better than that, but also like having a knack for 3-dimensional structure does take time, and we have learned a lot in this class. So if you don't want to spend that time and be comfortable with it, then this way is perfectly fine for drawing your peptide. I don't care about this section at all. So anytime we talk about a class of molecules, the book tries to give some sort of connection to that. Now, all peptides are important because for some reason or another, they're going to be incorporated into a protein. Some of them aren't incorporated into proteins at all. They just have little jobs all by themselves. So these small enkephalins are small 5 amino acid long peptides that act as painkillers inside the brain. But that is not something that you ever need to memorize. It's just something that the book teaches us about to give us a connection to those small peptides that we just learned how to draw. All right. So here is one of those pentapeptides. And here's another. So if we count our amino acids, I think it'd be kind of a cool question on an exam to say, you know, if you didn't know the sequence, if I didn't give you the sequence here, how many amino acids are in this peptide? And then you would have to actually count. So ammonium, alpha carbon, carbonyl. So that's 1 amino acid, ammonium, alpha carbon, carbonyl. 2 amino acids, ammonium, alpha carbon, carbonyl. 3 amino acids, ammonium, alpha carbon, carbonyl. 4 ammonium, alpha carbon, terminal carboxylate. That's the 5th amino acid. So that's a pentapeptide. All right. You can, even in a big molecule like this, you guys have the tools now to be able to look at all the parts of this and understand its reactivity. That's pretty awesome. So you can see in this side chain from the tyrosine that we have that polar hydroxyl group. So we know that that's a polar side chain. Phenylalanine here, all hydrocarbon, a non-polar side chain. The great thing is we've spent a lot of time on the organic chemistry part of the course in preparation for this. So when we see a peptide like this, it's not a shock to us. We can look at those side chains and sort of evaluate what their chemistry is going to be like. All right. What side chain is this one? Oh, wait, it's labeled. Sorry, I was trying to ask a question. The answer is already there. So that's leucine right there. There are also slightly more complex peptides that are cyclic, all right? And they can be cyclic in lots of different ways. One of the ways that we form the ring is by the covalent bond formed between two cysteine residues with their cysteine sulfurs. We call that a disulfide bond. So these are both nona peptides, nona meaning 9 amino acids long, and they are cyclic. They have very potent biological functions. They're different from one another. I happen to know of cyclic peptides that have different functions as well, because some of the most potent mycotoxins, the poisons that come from mushrooms, are also cyclic peptides, and they have incredibly potent effects on especially liver cells.
What would be the magic mushrooms like biological portion of it?
So psilocin and psilocybin, if you look at the slides from the amines chapter, I think psilocybin actually shows up in the structures there. So it has an amine functional group as part of its structure, but you can see its structure. And psilocin is a very similar molecule, but just hasn't been completely transformed into the slightly more psychoactive psilocybin. But they're amine functional groups in those. And the structures are in chapter 18. Psilocybin can be chemically synthesized. It's just really expensive to do it. And it's a Schedule 1 narcotic. So people don't tend to want to make that stuff because it would attract undue attention from law enforcement. It's a lot easier to just grow magic mushrooms in your basement instead. I'm not advocating for that, although I will suggest that magic mushrooms are by far the safest of the recreational drugs, even safer than marijuana, all right? The big danger with magic mushrooms isn't the mushrooms themselves, it's the possibility for misidentifying a mushroom if you're trying to harvest them, because magic mushrooms look very, very similar to mushrooms that are deadly toxic. So specifically mushrooms from the genus Galerina look very similar to the untrained eye to mushrooms from the psilocybe genus that contain psilocybins. So mushrooms are one of my passions, not taking them recreationally, but identifying them and actually foraging for edible ones. I was
gonna say, don't lie. You're out there going through the
no, I've
fields.
never I'm really boring when it comes to drugs. I freely admit that I've had too much to drink a few times, and beyond that, that's about it. That's all you got me for. So not that I think that other people that choose to expand or alter their consciousness in other ways, that's not a valid expression for them. I'm not judging anybody else. I'm just telling you that that's not my bag, personally. If I were to ever try a recreational drug at some point in my life, it would probably be some sort of psilocybin containing mushroom. So, but I haven't. I've never tried it. I found them several times, just if you're walking around looking for mushrooms in the Pacific Northwest, especially in the fall, you're probably going to run into psychotropic mushrooms at some point.
Isn't it usually right after the first frost?
Yep.
Then they all really pop up all breeds.
Psilocybe cyanescens and azurescens both pop up right after first frosts. Yeah. Mulch beds are a good place to find them. So there are lots of reasons why I don't recommend that novices go hunting for magic mushrooms. One of the big ones is that all of the habitats that psilocybes use Those deadly mushrooms, the Galerina, also use those same habitats. They're both types of mushrooms that are called saprobic mushrooms, which means that they grow on detritus. They break down dead wood, essentially. So growing on mulch is a habit for both the psilocybin containing mushrooms, psilocybin containing mushrooms, or Galerina, which is deadly toxic. So Galerina is not the only deadly mushroom, but it is one that looks to the untrained eye just like the psilocybe mushrooms. So go back to chapter 18 and look at those structures, Orlina. You can see psilocybin for yourself.
On it.
Yeah. So if we look at these two nonapeptides, this is not something you have to memorize anything about. I'm not going to ask you questions about these, but these are small peptides. A nonapeptide is only 9 amino acids big, and they've got important signaling jobs. All right. So oxytocin stimulates contraction and is used to induce labor. Vasopressin signals to retain water in environments where you might become dehydrated. Interestingly enough, those two look very, very, very similar to one another. Only 2 amino acids different between them. And really, isoleucine and phenylalanine, they're not all that different. Isoleucine and phenylalanine are both nonpolar side chain amino acids. Arginine and leucine are both nitrogen-containing basic amino acids. So these are incredibly similar molecules, oxytocin and vasopressin, but just those small changes are enough to give them incredibly different signaling jobs. Where are we at in terms of time? Oh crap, we're at an hour and a half. This is actually probably a great stopping off point for the night. So I will publish all of the videos for chapter 21 so that you can watch through everything. I think it's like an hour worth of stuff total. But we'll pick back up in recitation on Wednesday and talk about the different levels of structure within proteins, primary, secondary, tertiary, and quaternary, and then a little bit more about how the structure of the amino acid side chains influences how proteins fold. And I'll give you a little bit more information even than the book gives you because it's interesting stuff. It's worth knowing. So any questions? I promise the 21 videos will be up tonight. So it's just internet traffic hasn't let them process and upload. I have them like in process on YouTube right now. So I'll put links up, and as soon as you can access them, then take a watch. Remember, Friday's our exam too. Don't forget about that. Anything else? Well, thank you for coming and hanging out with me, Candace. That's nice. You don't have to thank me for doing my job. I like hanging out with you guys. I wish we were in class together. It's a lot better in class, trust me. So I'm still learning about how to be an effective professor online. And if you don't already have all of your content canned for online teaching, like I don't know, it's a lot harder to do this than to come and hang out with you guys in class. And it's a lot more chill. We're just, plus, like, you guys don't get to smell what an ester smells like. We can't do the lab where we synthesize those and try and get to guess, what those structures mean in terms of their sense. So I feel like you guys are missing out on some stuff, and I feel like I'm missing out because I don't get to meet all of you in person, and it sucks. So hopefully by next fall, you guys can come by the office and say hi. If you want to come hang out the week that we do our ester synthesis lab, I would not put that past any of you. So come by and do it if you can. All right. I'd love to have you as guests.
For this week, are we going to have a lab or not because of the test?
I think I actually do have a lab scheduled, and
Okay.
I actually have to look at that, Orlina. I don't know off the top of my head. Okay.
The next lab is very short and fast.
Yeah, I think that
Look at you.
I hadn't looked at it in a little while. I think that I did that on purpose because we had an exam week coming up. So I tried to put a lab in that wasn't going to be a pain in the butt. So
Yeah,
just do the lab. It's like, I literally, like, it'll take you 15 minutes to get it done.
I think it took like 10.
Okay.
Assuming your computer and program are operational, it's like 10 minutes.
Yeah.
Yeah,
it's-
that's pretty much, that's my goal for tomorrow, is do the lab and then start some of the homework as I go through the videos.
we could all use a few easy points at this point in the quarter. So that easy lab, like I'm throwing a bone to you guys.
Appreciated.
All right, if there's nothing else, then I will see you guys on Wednesday. I'll post an announcement when the 21 videos are finally finished processing and drop, so you don't have to be like,
Refresh.
yeah, don't refresh. As long as you have Canvas set up to give you alerts when announcements happen, you'll know when they're ready.
Perfect.
All right, have a good night, everybody.
Thank
All right.
you.